go ahead and get started. So today we are working on renal physiology, which is the last lab in our lab manual. Next week we have on the schedule a digestion lab, so I'm gonna get in touch with Professor Smith and see what he wanted to do with that. But as of now, this is gonna be the last one that's in our lab manual. I'll let you guys know what we're gonna be doing for lab next week once I find out from Professor Smith later on this week. But for now, we're gonna be working on renal physiology. We're gonna be taking a look at the kidneys, how we're gonna be producing our pee, and what our pee is gonna be telling us. So, is everyone able to see the PowerPoint?
Yes.
All right, good. All right, so today we're going to be taking a look at how we would be able to do what's called a urinalysis. Okay, and so what the urine is going to be telling us. Okay, so before we get into kind of the nitty gritty with this, just tell me right off the bat, what are some things you can tell just by looking at someone's pee?
Color.
Hydrated or
So
not.
you can tell color? And then I heard, I think, Karina, you said hydration?
Yeah.
Yeah, okay. What else can we tell?
The clarity.
Okay, so clarity. Okay. What else?
The amount.
Okay, we can tell the amount. So the volume, okay. All right, what else can we tell? What can the urine tell us? Anything else?
Is it just by looking at it?
Either just by looking at it or by analyzing it, just by, you know, any one of those.
If it's normal, not.
You can test the pH.
Okay, what do you mean by normal?
Testing.
Okay, so I have pH, okay.
Yeah, that's a good one.
Marina says edema. Anything else?
You can check like lab values like sodium and stuff like that.
Right, okay, so you can look for different ion levels, right? We can tell sodium levels and other ions. Okay. Kind of in that same vein here, looking at what's in the pee, what else can we find in our urine that can give us an indication about someone's health?
Blood.
Blood, okay. Okay. Right, anything else? Anything else? What was that? What was
Kidney
that?
stones. What is it? Kidney stones?
Couldn't quite hear you. Say that one more time for me.
Kidney stones?
Kidney stones. Okay. We can also find glucose, right?
Yeah.
So we can find a bunch of different substances inside the urine that are going to give us a different side of the picture about someone's health, right? So we can look at color, and that can tell us about their hydration. We can look at the amount of urine that they're producing. We can look at the amount of salt or the amount of other solutes or glucose. We can find kidney stones. We can find blood. We can find a whole lot of different substances. In some cases, we can even find drugs. And so all of that can give us an indication of what's going on inside of a person's body. So today, what we're going to be doing is we're going to be taking a look at how the kidneys are able to show us that big picture. So that's going to be our goal for today here. Okay. So when we start by taking a look at the urinary system, and so the urinary system is going to be comprised of two kidneys, okay, one on either side of the body. And coming out of each kidney is going to be a ureter. The ureters will ultimately lead into the bladder, which will be storing the urine. Okay. And then the urethra, which will be the pathway that the urine is going to be taking to get into the exterior of the body, ideally into the toilet. Okay. So that's going to be our urinary system. And so the goal of the renal system or the urinary system is to be able to maintain your blood homeostasis. Okay? You want to keep everything in the blood at an appropriate level. If any one of those levels gets kind of out of whack, okay, you're going to be able to drop off, you know, any extra substances into the urine to get you right back down to your normal set point. Okay? So the way it's going to be doing that is the kidneys are going to be filtering the blood. And as it filters the blood, it's going to end up returning most of those substances back into the blood. Okay? So most of the water and most solutes that get filtered are going to get returned back into the blood. Okay? Anything that doesn't get returned to the blood is going to be made up, or is going to be kind of comprising your urine. So any extra water that doesn't get reabsorbed, or any extra solutes that don't get reabsorbed back into the blood will make up your urine. And again, the goal of creating that urine is to keep your blood at an appropriate, or all the levels in your blood at an appropriate level, and maintain your blood homeostasis. So to do that, to maintain your blood homeostasis, your kidneys are going to be able to do several different things. Okay. Number one, it's going to be able to regulate your ionic compositions inside the blood. So I think Brittany had mentioned earlier, looking at how you can regulate your sodium levels, your potassium levels, and chlorine levels. And those are going to be the major the major ions that the kidneys are going to be responsible for regulating. Okay. So for example, if your sodium levels get too high in the blood, then what the kidneys can do is it can throw more of that sodium back into your urine, okay. And then you end up peeing out that extra sodium, and you bring your sodium levels back down. Okay. Same thing with potassium, same thing with chloride. Okay. If those levels are too high inside the blood, you just pee out whatever you don't need, and then that helps you to bring your ion levels to an appropriate set. Okay. So that's number one. Okay. Number two is it's going to be able to regulate your blood pH. Okay. So remember, your blood pH should be about 7.4. Okay. If your pH is too acidic, so your pH is a little bit lower than that, what your body can do, or what your kidneys can do, is it can excrete extra hydrogen ions. Okay. So as you excrete extra hydrogen ions, that's going to be fewer hydrogen ions getting pushed back into the blood. More of that is going to be pushed into the urine. And as a result, you're going to end up bringing your blood pH from a lower level back up to 7.4. So again, the goal is to help to excrete the extra acid in order to bring your sodium level, or your pH back to a normal level. Now, the flip side of that is if your pH gets too high, then we can start to excrete bicarbonate. So if our pH gets to about 7.5, for example, then we can excrete extra bicarbonate. Okay, that's going to be that base that we're going to get rid of, and that's going to help us to bring our blood pH back down to 7.4. So we're going to be able to regulate various different ions for different purposes. Okay? In addition to that, we're also going to be able to regulate our blood volume. Okay? So if we consume too much water, and let's say we're just drinking water, okay, we're going to end up with more water inside of our blood. Okay? And so as a result, that's going to cause the blood volume to increase. Well, the problem with increasing your blood volume is your blood vessels are not increasing with it. They're going to be about the same diameter, okay? But they're filling with more blood. So what that's going to cause is it's going to cause the fluid inside the blood to press more on the arteries. And as a result, that's going to increase the blood pressure. And we don't want to have an increased blood pressure. We don't want hypertension. Okay? So if we have increased blood volume, again, what we can do is we can just pee out that extra water and reduce our blood volume. Okay? And then now we have an appropriate blood pressure. So by doing all of that, it's going to be able to regulate your body's blood osmolarity. Okay, so osmolarity is the number of solutes dissolved inside a certain liter of solution, or per liter of solution. In this case, our blood. Okay. So this will tell us the amount of particles dissolved per liter of blood. Okay. And we want to maintain that at an appropriate level and keep that regulated. Okay. The last thing that we're going to be able to do is we're going to excrete any waste. Okay. So waste can be any excess substances or any excess solutes. They can be anything that does not serve any purpose to the body. Okay, so for example, like any certain medications are going to get excreted out through the urine. Alcohol is going to get excreted out through the urine. Drugs are going to get excreted out because the body doesn't really have use for that stuff. And any foreign substances like bacteria or anything like that. All those substances that we do not want inside of the body have the ability to be excreted out as urine. So these are all the different functions of the kidneys. And again, the whole goal of this is to regulate your body's blood homeostasis. Okay. Any questions on any of these so far? Okay, cool. All right, so that is that. Okay, so we know the kidneys have to be able to regulate the blood composition, right? So how is it going to go about doing that? Well, what's going to happen is most of that blood, about 20% of your blood volume, is going to end up running to the kidneys through the renal artery. Once it enters the renal artery, it's going to filter, or it's going to that artery is going to divide into smaller arteries and eventually circle around some nephrons. And so in the kidney, we have about 1 million nephrons, and the roles of the nephrons is to actually do the filtering of the blood. So these are the actual components that are of the kidneys that are actually doing the work here. Okay. So all of that blood is going to get filtered specifically inside the nephrons of the kidney. So this is just kind of a basic picture of what the nephron would look like. Have you guys talked about the kidneys already in lecture? Yeah, I know.
Honestly, I'm not sure where he is with lecture.
Oh, okay. And have we talked about the kidneys or the renal anatomy or physiology? Anybody? Okay.
I'm pretty sure he has.
So, okay. All right. So if he has, I'll just kind of go over it in a little bit of extra detail here just to make sure we're all on the same page. But the goal, this is just kind of a basic overall kind of schematic of what's going to be happening inside the nephrons. Okay. So typically what's going to be happening is we're going to have some fluid, our blood running through our glomerular capillaries. They're going to enter the glomerulus here. And inside the glomerulus, most of the blood is going to get filtered out. So most substances inside the blood are going to get removed from the glomerulus, from this blood vessels here, and they're going to move into the renal tubule, specifically into Bowman's capsule. Okay. So all the substances that get filtered out are what we refer to as the filtrate. Okay. Now, that's going to be the first process that's going to happen inside the nephrons. Okay. The second process that's going to happen is what's called reabsorption. Okay, so reabsorption is going to be where we reabsorb about 99% of the filtrate that we just filtered out. Okay? And we're going to reabsorb it back into the blood here. Okay? So this is where we have reabsorption. The majority of that reabsorption is going to be taking place specifically in the proximal convoluted tubule, as well as in the loop appendage of the nephron. That's going to be where most of the reabsorption will be taking place. Okay? So after the substances get reabsorbed, or certain substances get reabsorbed, anything else that was not able to get filtered out before is going to get secreted from the blood into the renal tubules. Okay? And then whatever comes out of the renal tubule after this process becomes the urine. Okay? So whatever gets filtered out, plus whatever gets secreted, minus whatever gets reabsorbed, will be whatever becomes excreted as your urine. Okay? So those are the four processes. Okay? We're going to have filtration inside the glomerulus. We're going to have reabsorption, okay, anything that we want to retain inside the body. Okay? Anything that we want to get rid of, okay, we're going to go through secretion and secrete it from the blood into the renal tubule. Okay? Whatever comes out of this process, or whatever's left over after this process, I should say, becomes your urine. Cool. Any questions so far? Does this all make sense?
Yes.
Yeah, okay. Okay, cool. All right, so that's going to be how the kidneys are going to be able to filter blood. Okay? And so now when someone actually goes to the bathroom and goes, to pee, what we can do is we can look at the composition of the urine, and the urine can tell us a lot of different aspects of someone's health. Okay? And tell us specifically about the health of them overall. It can tell us about the health of specific organs or specific organ systems. Okay? And so a few things that we want to really be keeping track of are some casts, some crystals, and some bacteria. Okay? So if there are any casts inside the urine, the casts are just these proteins that lie inside the urine. Okay. Now, typically, proteins should not be ending up in the urine because they're too large of molecules to get filtered out. Okay. But if proteins are getting pushed into the urine, that is usually an indication that someone is dealing with hypertension, or they're dealing with glomerulonephritis, or there's a problem or inflammation of the glomerulus itself, or some other problem. Okay. And so that can be a way that we can indicate either cardiovascular health or kidney health. Okay. The second thing that we're going to look at are crystals. So crystals, kind of like kidney stones. These, again, should not be inside the urine, but if we do find them, then again, that can tell us different aspects of that person's health. And then the third thing that we're going to be looking for specifically is going to be bacteria. And so if there's any bacteria inside the urine, that can be an indication of a couple things. Okay? Either one, they have a UTI, so they have an infection inside the urine, and you're peeing out some of that bacteria that's causing the UTI. or you may just be peeing out any bacteria that are found near the urethra. Okay? So if there's any bacteria near the urethra, as you pee it out, that bacteria might get caught inside the urine stream and then end up inside your urine. Okay? So those are the kind of the three major kind of things we're going to be looking for inside the urine, as well as a number of other things, which we'll look at in a second. Okay. So this is a table that's inside your lab manual. And so these tell you kind of what the normal values of each of these different properties are and what the abnormal values may indicate. Okay. So for example, when we're looking at the color of the urine, it should be running from either a pale yellow color to a deep amber. Okay. If it's anything other than something in that range, that can lead to a number of, or that can kind of indicate many different problems inside the body. Okay. Typically, there shouldn't be an odor to the urine, but if there is, that it can either lead to, that can either indicate dehydration or someone who's dealing with diabetes. If there's any white blood cells inside the urine, again, that's going to be an indication there's going to be a UTI. The white blood cells are going to be there to try to fight off that infection. If there's some proteins in there, that can be an indication of hypertension because, again, the blood pressure is too high and pushing the pushing the proteins into the urine, which, again, shouldn't be happening. It can be an indication of glomerulonephritis or eclampsia. pH should be about anywhere from 4.5 to 8, which, again, will indicate kidney dysfunction, or it can just indicate someone who's got an unusual diet. There shouldn't be any blood, but if there is, that can be because of a kidney infection or a bladder infection. The specific gravity should be at a specific range, and we'll talk about what that means in a second. And there shouldn't be any glucose. Okay. And so if there is any glucose, that's going to be an indication that someone is dealing with diabetes or possibly stress. Okay. And so, yeah, those are different types of properties, okay, that may be found inside the urine that can be used to kind of piece together the puzzle of what a person's health is looking like. Okay. Actually, before we go forward here, kind of fun fact. Does anyone know how diabetes used to be tested for, like way back in the day? Does anyone know how people used to test for, or doctors used to test for diabetes?
They used to drink it.
Yes, they used to drink it. Right. So way back in the day, before we had all these different types of urinalysis techniques, what doctors would do in order to test for diabetes is they would taste their patient's urine. Okay? And the urine tasted sweet. That was an indication that there was lots of glucose inside the urine. And so that was how they would diagnose diabetes. So thankfully, we've come a long way from that. And so we were able to just do some tests to help us identify diabetes, and we don't necessarily need to drink our patient's pee in order to determine that. So we've come quite a ways from that. So that's good there. And then again, we can have some crystals. Crystals can indicate gout or kidney stones and things like that. Bacteria would indicate a UTI, things like that. So there's all kinds of different properties that are going to be useful for telling us what a person's health is looking like. All right, so these are some pictures of some crystals that you might see in someone's urine. And again, you can kind of see how kind of like actually crystalline they actually look. And so this is what we would see if we were looking at someone who had kidney stones. And then here's another one as well. Okay, again, you can see some red blood cells along with the kidney stones in someone's urine, okay, which again, you don't want to be seeing. So this guy's got all kinds of problems here. Okay, so another thing that we want to be taking a look at is the volume and the volume of the urine, as well as the salt concentration of the urine. And so volume and salt concentration inside the blood are going to be regulated by specific hormones. Okay? The two hormones in question here are going to be antidiuretic hormone and then another set of hormones involved with the renin-angiotensin-aldosterone system, or the RAS system. So the first one is antidiuretic hormone, or ADH. And so what ADH does is it will increase your water reabsorption. Okay? So it's going to allow you to be able to reabsorb water back into your blood and help you with water retention. Okay? And so this is typically going to be found in someone who is dehydrated. So if someone's dehydrated, what they want to do is they want to retain as much water as possible. So their body is going to release lots of ADH to help them bring that water back into their blood and help them kind of retain their blood volume. Okay. Now, fun fact about this is also the hormone that's caused or that leads to the phenomenon we know as breaking the seal. Okay? So have you ever been out partying with some friends, or you're out at the bar on St. Patty's Day or whatever it may be? You might be having a good time, having some of the happy juice, okay, drinking quite a bit. And then you start to realize, after maybe an hour or two, that you have to go to the bathroom. Okay? So you go to the bathroom, you go pee, you come back, you start drinking some more, and then maybe every 30 minutes or so, you start realizing you have to go to the bathroom over and over and over again. Okay? Well, the reason for that is that alcohol actually inhibits anti-diuretic hormone, so it prevents you from releasing anti-diuretic hormone. Okay? Now, if you're just drinking and drinking and drinking, you inhibit your body's ability to produce anti-diuretic hormone, and you just constantly have no way of reabsorbing water. Okay? So anything that you drink is going to end up directly inside your urine, and you're just going to pee over and over and over and over. And so with every single alcoholic beverage that you have, you inhibit the ADH more and more and more. So by the time you realize you have to go to the bathroom, you've already inhibited it so much that all you're going to be doing for the rest of the night is not having any water reabsorption, and you're just going to be constantly peeing throughout the rest of the night. Okay. Does that make sense? So that's kind of the hormone behind breaking the seal. Okay, so that's that. The second kind of system here is the renin-angiotensin-aldosterone system, or the RAS system. This is involved with your salt reabsorption. Okay. So if you're in a situation where you don't have as much solutes inside your body as you should, the RAS system will kick in and help you reabsorb some of that salt. Okay. And that's going to help you to maintain your blood pressure, or keep a high level of blood pressure, and then keep your blood solute concentration at an appropriate level. So both of these are going to help you regulate your water and salt reabsorption inside the nephrons. Okay, any questions on what's going on with ADH or the RAS system?
No.
Do these guys make sense? Yeah, okay. All right. All right, so when we're looking at water, one thing that we can do is we can be drinking large amounts of water, right? So if we're drinking large amounts of water, what that's going to do is it's going to increase our blood volume, because again, we're drinking more water, the water is going to end up inside of our blood. Okay? So if our blood volume begins to increase, okay, that's going to cause our blood to become more dilute, okay, because there's just more water in there and not as high of a concentration as solutes. Okay? So the blood becomes more dilute, and so what we do here in order to kind of combat this is we reduce the amount of ADH that we produce. Okay? So we don't produce as much ADH, and so as a result, you don't reabsorb as much water, and more of that water is going to stay inside the nephron and then ultimately get excreted out. Okay? And so that's going to be a way that you can kind of regulate your blood volume and keep it at an appropriate level. Okay? And so as a result, what's going to happen is you're going to be producing a more dilute urine that's going to be pretty clear and pretty light. That's an indication that someone is pretty hydrated. The flip side of this is if someone is consuming large amounts of salt, that salt that we consume is going to increase our blood osmolarity. Okay? And so as a result, we're going to have a lower water concentration compared to our salt concentration. Okay? So in order to combat this, our body is going to release a number of different types of hormones, and that's going to help with water reabsorption. And so as we reabsorb our water, that's going to help to kind of balance out these salt and water concentrations, again, to get it back to an appropriate level. Okay. Now, increasing the water reabsorption through this process is going to result in a smaller volume of urine, because you're not going to be excreting as much pee or as much water. Okay. And so the urine that you do produce is going to be pretty concentrated, because again, you're going to be peeing out more of that salt. Okay. And so that's where you would end up with a darker urine. All right, so one of the measurements that we want to be able to take in a urine setting here is what's called the specific gravity. Okay. So specific gravity is a measure of the solute concentration inside someone's urine. Okay. Now more specifically, it's a measure of the density of the substance that you're looking at, in this case, the density of the urine, compared to a standard. And when we're looking at urine, the standard is going to be water. Okay, so water will typically have a density of about 1.0. Okay, and the urine is normal urine should have a density or a specific gravity of 1.01 to 1.03. Okay, at a normal level. So it should be slightly more dilute than normal water, or sorry, slightly more concentrated than normal water. Okay. Now, if our specific gravity is higher, okay, that's going to indicate that there's more stuff inside the urine. There's more solute inside the urine, okay, which means there's a higher concentration of solutes. Okay, and if you have a higher concentration of solutes, you're going to have a lower concentration of water inside your urine. Well, if you have a lower concentration of water inside your urine, you have less water inside your urine, that's going to indicate that that person is less hydrated. Okay? Which again makes sense because if you have less water inside the urine, okay, there's not going to be as much water inside your body. Okay? So that's going to indicate that someone is going to be less hydrated, and this is typically where you'd see someone who has a darker urine. Okay? The flip side of this is if you were looking at someone who has a lower than normal specific gravity, okay, that's going to indicate that there's a lower solute concentration inside the urine, which means that there's going to be more water inside the urine. Okay? If there's more water in their pee, okay, then we know that the person is pretty hydrated. Okay? And so they would have a more clear urine. Okay? So a higher specific gravity means that the person is less hydrated. A lower specific gravity will tell us that the person is more hydrated. Okay? And so the way you would measure specific gravity or calculate specific gravity is you would take the density of the substance, in this case, the density of the urine, and divide it by the density of the standard, in this case, would be water. The urine would, or the units would cancel out, and whatever number you would get is your specific gravity. And you can use it to determine the person's hydration level or the amount of solutes inside their, or the relative number of solutes inside their urine. Does this one make sense? This is a pretty big concept here. Any questions on specific gravity? Okay. All right, looks like we're good. Okay, so when we're looking at specific gravity, this is going to be really, really helpful when we're trying to diagnose different types of diabetes. So the more common diabetes that we're more familiar with is called diabetes mellitus. That's the one where people's insulin either is not being produced or it's not being as efficient. Okay? And so if insulin is not being used efficiently or if it's not being produced, that's going to lead to a situation called hyperglycemia. Okay? Hyperglycemia just means you have extra glucose inside your blood. Okay? And so what happens when that blood starts to filter through the kidneys is that the glucose enters the urine a lot faster than it's able to be reabsorbed. Okay? And so as a result, as a result, you're going to end up having more glucose inside your urine, okay, because you're not able to reabsorb it fast enough. Okay? And as a result, you'll end up with a higher specific gravity. Okay? So you have more glucose inside your pee, which causes you to have a higher solute concentration, leads you to have a higher specific gravity, higher than normal specific gravity. Okay? The flip side of this is diabetes insipidus. So diabetes insipidus is where someone does not produce ADH as well, or their ADH is not as efficient. Okay? And so if you're not producing any ADH, okay, then you're not going to be able to reabsorb any water. Okay? So if you're not able to reabsorb water, you're going to end up with more water inside your urine, and then you're going to have a more dilute urine. Okay? So you'll have a lower solute concentration inside your pee, and as a result, you'll have a lower specific gravity. So the specific gravity can be used to help you diagnose whether someone is dealing with diabetes mellitus or diabetes insipidus, or if they have a normal glucose level or normal health. Okay. All right. So we kind of looked at all of the different factors that we're going to be looking at inside the urine. And so the way that we would typically be doing a urinalysis is by looking at one of these three processes. Okay? So urinalysis is going to be a process where you analyze the volume of the urine, the physical properties of the urine, the chemical properties of the urine, and even some microscopic properties of the urine. Okay? And so this can be done in one of three processes. The first process is what's called a complete urinalysis, where you take someone's urine, you put it under a slide, and you look at it under a microscope, and you look for different microscopic components that are inside the urine and use that to help make a diagnosis. You can do what's called a rapid urinalysis, where you have someone pee in a cup, and then you take a little urine test strip, you dip it inside the urine, and all these little pads will turn a specific color. And each one of these pads will give an indication of a different characteristic about the urine. You might have glucose for one, you might have pH for another one. Another pad might represent certain like casts or proteins that may be present, or blood that's present, and so on and so forth. Okay. So you can do a rapid urinalysis. This happens, is able to occur very, very quickly. Or you can do what's called a 24-hour urine collection and have someone pee in a cup over a 24-hour period. And over that 24 hours, you're able to analyze the urine each time, and that'll give you a more holistic picture of that person's overall health. And so by looking at what's actually inside the urine, we're able to determine if anything that's normal is present or anything that's abnormal is present. Okay? So if there's anything that's abnormal, that's going to be an indication of different diseases or conditions that that person may be dealing with. And then we can kind of go about trying to come up with a solution to help treat that, treat that patient. Okay, so that is it for today. So are there any questions on anything we've talked about so far?
No.
No? Okay.
No.
All righty. So if we were in lab today, we would have the opportunity to do a urinalysis. Again, unfortunately, we are not so lucky here. So what we would be doing in lab is we would be having a few people in our groups peeing in a cup over a 2-hour period, and we would go through and analyze their urine, which is a pretty, as gross as it sounds, it's actually a pretty fun lab. But unfortunately, we got to kind of do something different. So for this week's lab, we kind of ran a little simulation here. And so here we have a physiology class that's undergoing, that's doing a urinalysis study. We have 4 individuals, okay, who pee in a cup at time 0. That's going to be the first time they pee. And after that point, they're going to start consuming 1 of 4 different substances. Okay. So subject 1 is going to be consuming water. Subject 2 consumed Gatorade. Subject 3 drank some Mountain Dew. And then subject 4 consumed some very salty pretzels. Okay. And so after they took that initial sample, they consumed whatever food or drink they're supposed to be consuming. And then we look at the measurements, specifically of volume, specific gravity, and salt concentrations at different time intervals, okay? At 30 minutes, 60 minutes, and 90 minutes. Okay? So here are the graphs for each of those different characteristics. Okay? So here we have the urine volume that they produced. That's the first graph. The second graph is a specific gravity, again at time 0, 30 minutes, 60 minutes, and then 90 minutes. Okay? That's the specific gravity of their urine. And then the third one is their salt concentration. Okay? Again, at time 0, 30 minutes, 60 minutes, 90 minutes. Okay? So what you're going to do first is you're going to use these graphs to kind of fill in these tables. You're going to list the volume of urine that they produced at the 0-minute mark, 30, 60, and 90 minutes, okay, for each substance. Do the same thing with the specific gravity, and then same thing with the salt. Okay, so you're going to do that for the person who drank water. Okay? Then you're going to do that with the person who had the pretzels, the person who had the Gatorade, and then the person who had Mountain Dew. Okay. So you're going to fill in that table using the graphs here. So it should be pretty straightforward. Everything is there for you. You're going to enter in those values, and then you're going to answer the questions here. Okay. So the first question asks you to compare the results in the tables and explain the results between the water drinker and the Gatorade drinker, the water drinker and the pretzel eater, and then the water drinker and the Mountain Dew drinker. Okay, compare the results and see if you can explain why we saw specific results. And then the second question asks you why an animal with a longer loop of Henle would produce smaller volumes of more concentrated urine. Okay? So that's pretty much it for this lab. You're going to answer those questions. Once you're done, you're going to turn them in on Canvas, and then you will be good to go. Okay.
I have a question.
Any questions? Yes. Yeah, go ahead, Karina.
We go back to the specific gravity part. Are we just going to average or like kind of guesstimate because it's to a tenth of %?
Yeah. So just kind of estimate on it. It doesn't need to be specific. Just doesn't need to be like a specific actual like hard and fast number that's accurate. As long as you give me like an estimate, that's totally fine.
Okay.
Yeah. Go. Any other questions?
Where do you post the recordings from the Zoom?
Where do I post? Sorry, say that one more time.
You record our sessions here. Do you post them?
Yes. Yes. So I'll go ahead and show you right now. Let me go over.
There's just a lot of information, so I think I need to like hear it one more time.
Yeah, it is a lot of information. So if you go to the normal like lab page or your Bio 9 Canvas page, the lectures are all posted underneath that particular topic. So this is the Lab 14 one. So under Lab 14, you have the Lab 14 pre-lab lecture. This will take you to the YouTube page for that particular lecture.
Okay, I got it. Thank you.
Yeah, no problem. And so the next one will be uploaded right here, either tonight or tomorrow. Cool. All right, any other questions?
Can you just go back to the page and maybe like show us like one, like how you would, like, I guess I get confused when you have to like average it.
Okay, so what we'll do here then. So here is, okay, we'll do the volume, for example, right? So we're gonna look for subject 1, subject 2, subject 3, and subject 4. So subject 1 is the one that's in blue. Okay. So their initial volume is about 30 milliliters. Okay. So that's gonna be at time 0. So you're gonna go over to the water. At time 0, you're gonna just write in 30 milliliters.
Okay, so literally just average it out.
Yeah, you're literally just straight writing the numbers in from the group.
Okay.
Yeah. Nothing special is going on here.
Okay.
Same thing for specific gravity. Here we can kind of, it's kind of in the middle of 1.01 to 1.02. So we'll say this is 1.015 for the specific gravity at time 0.
Okay.
And then the salt concentration, let's say that's about 12, and you just put in 12.
Okay, cool.
So you're literally just going to do that for all of the different numbers and values here.
Okay. And then George, are we take those numbers and then kind of look at our chart for the test and readings and kind of figure out what it is that we're looking for on the color chart?
Yes.
That chart?
Yeah. Wait, sorry. Ask your question one more time. I think I understand what you're saying, but I'm not sure.
Okay, so for the discussion questions, we have to explain our results. So is it, were we supposed to look at the test and readings, like the different colors for the multi-stick chart that's in our lab?
Yes. So,
so the colors, but from our lab report, you can't. Like the print from our workbooks, you can't really tell with the color chart, but from your PowerPoint, the colors are there for us for like glucose and, all the different readings and the colors.
Oh yeah, so don't worry about that. Don't worry about that. You won't need any of that.
Okay.
All you'll need are just the values here. Yeah. All right, cool. Any other questions?
Off topic, when is our lab final going to be?
The lab final is scheduled for, let me look at it real quick. The week of May 12th. So that would put us, so May 12th. So 2 weeks from today.
Okay.
So I will have more information on that for you this week. I'm not sure exactly how Professor Smith wants me to go about doing that just yet. So once I have that information, I'll let you know as soon as I can. Okay. So you have enough time to prepare for that. Yeah.
Excellent. Thank you.
Okay. No problem.
Make it super easy.
So if there's no more questions, you guys are free to go. If you have any more questions, I'm not going anywhere. I'll be here.